So my talk will be about the fundamentals, the basic science about the photoacoustic phenomena and some applications. The photoacoustic phenomenon starts with the absorption of light. So after the light is being absorbed by a chromophore or a molecule, the excited states are deactivated by several mechanisms. One of is by heat, by deposition of heat, and in the singlet or in the triplet states. This deposition of heat is the main is the best case scenario for photoacoustics, because after heat is being deposited, a pressure wave will be formed. But there are other mechanisms. For example, fluorescence that does not contribute to photoacoustic signal, and there could be energy transfer to another molecule, such as oxygen, and all these mechanisms should be competing at the same time after light being absorbed. But anyway, even these three mechanisms are competing, there always be some heat being released in these vibrational states, a very fast heat deposition that will generate a photoacoustic signal. So this is good because we can study molecules that have fluorescence and molecules that are applied for phototherapies, for example, or they have another applications. But to work as a contrast agent, for example, the best case scenario is this of non-radioactive decays in blue here. So what is the conditions for efficient photoacoustic signal generation? One condition is the thermal confinement. That means that the release of the heat should be kept during the releasing time of in the volume of the laser where it is being absorbed without diffusing by the medium. So this depends on the thermal diffusivity and on the spatial resolution. The other condition is the stress confinement that depends on the speed of sound. That means when the heat is being released, the pressure wave stays confined in that volume and do not propagate through the volume. So these two conditions means that you should have a pulse laser with shorter time than the thermal confinement or stress confinement. So this is the best condition for photoacoustic generation. One example is if you have this thermal diffusivity of soft tissues, that's 0.14, we can get a thermal confinement of 18 milliseconds, and for this resolution of 100 microns, and stress confinement of 600 nanoseconds, more or less. So if you use a laser, a pulse laser of 10 nanoseconds, this is a very good condition, or fulfill these conditions. In these 10 nanoseconds is usually the lasers employed by the photoacoustic system. So how light converts to sound? Well, there is a relation between the fractional volume expansion related to the pressure and the temperature. So when these two conditions are achieved, that the pulsed light of the laser is shorter than the thermal confinement or the stress confinement, this volume fraction over the volume is near to zero. So we can write the pressure in function of the temperature. Well, the temperature can be written in function of the absorbed energy. And the absorbed energy can be written in terms of the absorption coefficient and the laser fluence. So we have the pressure wave in function of molecule property, that is the absorption coefficient, and in function of the laser fluence. So if you have a molecule that absorbs lots of light or have a high cross-section of absorption, it will increase the pressure or if you increase the laser intensity, it will increase the pressure too. So this is in function of the constants, like the thermal coefficient of volume expansion, the isothermal compressibility, specific heat, and so on. All these constants are gathered in the Gruneisen parameter. So in the end, you have the pressure in function of the absorption coefficient, that's a molecule property, and the laser fluence. And the constant is the Gruneisen parameter. This is a picture to summarize what I said before. So you have the tissue here, and you have here a spot that the contrast to be absorbed, or the molecule that you produce photoacoustic signal. So when it is irradiated, there is a local temperature rise, a very fast one. This is in the femto to picoseconds. Then this very fast heat deposition will generate a pressure wave that will be detected by ultrasonic transducers and reconstructed as an image. So in practice, how this works in practice? In general, we have a device that emits the laser and collects the pressure waves in the same device. So the device has a laser and has a transducer in the middle. So using only one device, we can irradiate the volume we need and collect the pressure wave back and reconstructed an image. Another thing about these photoacoustics is the tissue light absorption, because if you are talking about photoacoustic in vivo, for example, or for animals study, for biological studies, we need to consider the optical depth penetration. So below 600, more or less, the body or the biological tissues has several components or several molecules that absorbs there. So the light will not penetrate. So we should consider the photoacoustics above 600 nanometer, because we have more penetration of light and we can imagine more deep in the tissue. Photoacoustics relies on the endogenous absorbers. For example, we have the oxyhemoglobin and the deoxyhemoglobin that have different absorption spectra, and we can get a contrast of both of them. We have also water and lipids, especially in this peak here for lipids, that's 1,300 nanometers, more or less. So these endogenous absorbers, we can divide these in near infrared 1 region and near infrared 2 region, mainly because the equipment has 2 laser systems that is separating these 2 regions. As exogenous contrast agents, we can have organic dyes and nanomaterials, nanostructures dyes. These 2 general sets of exogenous compounds. For example, we have this bacteriochlorine that absorbs in the therapeutic window. And this one has this manganese metal in the center that will increase the non-radioactive decay. So it absorbs at 830 nanometers, and most of the light will be returned to the ground state as a non-radioactive decay, inducing a photoacoustic or producing a photoacoustic signal. Here we have a phthalocyanine complex that absorbs 850 nanometers, and that will produce a huge signal, photoacoustic signal too. So these 2 molecules are examples of photoacoustics exogenous contrast agents. It's more or less biocompatible too, because they are organic, they are more biocompatible. We have other examples like this bacteriochlorine that does not have a metal in the center, so this is used for photodynamic therapy, meaning that this molecule induce reactive oxygen species. But we can use photoacoustics to, for example, study the biodistribution of these molecules, or to study the accumulation of this molecule in the tumor, for example. So we can use photoacoustics to study these kind of molecules too. In the induction or production of singlet oxygen or reactive species of oxygen, of course, you have some effect, some collateral effect or side effect, but it's not too relevant, because the photoacoustic tomography is, in general, did not take too much time to collect an image. Another example is the indocyanine green compound that is used as a fluorescent probe. So part of the light will be lost as a fluorescence and not generate photoacoustic signal, but part of the signal will induce photoacoustic signal. So we can, again, study the biodistribution or study the accumulation and so on. Indocyanine green is interesting because in the monomer form, it absorbs here at 800, a little bit above 800, and it is a fluorescent molecule. But in the aggregated state, this molecule absorption goes to above 800 and does not fluoresce anymore, just generate photoacoustic signal. So all the light that's absorbed in this aggregate of indocyanine green is lost by heat. This is a set of contrast agents that are being studied, or families of contrast agents in function of the molar absorption coefficient. Thus, we have organic compounds here that have this molar absorption coefficient 10 to 6, 10 to 7, and then we have nanoparticles that have much more higher absorption coefficients. But this is a trade-off, because the nanoparticles are generally are not biocompatible. So organic compounds has lower absorption coefficient, but are more biocompatible, and the nanoparticles is the opposite. So it's a trade-off in choosing the contrast agent and what's better. This is how the system works, or some systems. We have fiber optics here that emit the light, and we have the transducer here. This is the equipment we have at the Coimbra Laser Lab. Our equipment, we can do ultrasound, normal ultrasound, like beam mode ultrasound. We can do a photoacoustic mode, and we can do Doppler mode. So we have 3 imaging techniques in one equipment. This is the laser, and this is the equipment, the software. And here we have the animal, where the animal is positioned, and we have this 3D motor that will move as we want. Here's some examples of imaging. This is just ultrasound and beam mode imaging of the liver of a rat. So it's a portal vein here, inferior vena cava here, and we can measure the size. We can do several measurements here. This is another example using a 40 megahertz transducers. As higher as the frequency, as better the resolution, but as higher the frequency of the transducer, less signal will reach, because it will be attenuated. So again, there is a trade-off here between the resolution and the transducer. So this image is the lymph node of a mouse leg. It's very well defined here. This is a photoacoustic signal merged with the beam mode ultrasound. We just imaging at 850 nanometers just to see the vessels distribution, how the blood vessels are distributed around the tumor. So we have a sequence of imaging. Here we can see the tumor. This is the tumor. we cannot see, there is no much vascularization inside the tumor, but we have lots of vascularization on the surface, okay? We can reconstruct a 3D imaging and merge the imaging of the blood vessels distribution and the anatomic given by ultrasound mode, and you can separate both and have just the blood imaging, the blood distribution imaging. Here's an example when you use this radaporfin. It's used for photodynamic therapy, how it distributes around the liver. So this is the liver imaging, and you have the compound here, distribution, and you have the spectrum of the compound. That's very important to have a spectrum because you can see if you are looking to the right compound or for detecting that. This is the same example, and you are detecting oxyhemoglobin, the oxyhemoglobin, and the radaporfin. That's the exogenous compound, and the distribution on the liver. This is an example of the Doppler for the cardiac studies. This is an example of the Doppler for the kidney. We can see, well, the Doppler, we see just the flux of the blood. If it's going forward or toward some place. So in this case, blue and red means that the blood is flowing from one side to another side, okay? This is the kidney. So we are looking at the vascularization of kidney. Here is another example. We are studying the permeation of some compound. It's a bacteriochlorine that absorbs at 730. This compound is used for topic applications. So we are seeing this in the mouse skin. So we measure this is 300 micrometer, sorry, 300 microns or 0.38 millimeters. And you can again make the spectrum to be sure that we are seeing this compound and not the blood or not other compounds. This is the Doppler for the brain studies. Again, blue and red means the direction of the blood flux. And this is a very nice example of photoacoustic application for Alzheimer's disease. All these neurodegenerative disease cause an increase of copper in the blood. So this increase in the copper levels are related to this neurodegenerative disease. So this group make a compound that does not absorb in this form without the copper. And when this compound finds the copper, it chelates the copper and starts to absorb. This is in green here. And starts to give photoacoustic signal. So it was injected in the mice. When this normal mouse that doesn't have Alzheimer's disease, there is no photoacoustic signal. In the mice that has the Alzheimer's disease, the contrast is very good. So it means that this compound crossed the brain barrier, a brain-blood barrier, and was able to chelate the copper and indicate that this mouse has Alzheimer's disease. And this is the control using just PBS. So we have a very, very nice contrast. This is more applications for inhuman, it's for ovarian cancer, and this is for breast cancer. In this case, they are just imaging the fat, water, or lipid and water and blood. So this is endogenous contrast agents. They are not using other contrast agents. This is the same. So we can get several types of contrast in the imaging, given to the colors. And this is similar, but for the carotid. Only looking to the lipids and the water and etc. So I finished my talk, and before I finish this, I would like to announce the International Conference on Photoacoustic and Photothermal Phenomena that will be held here in Coimbra on July 2024. So everyone is invited to participate. And to finish, I'd like to thank all the members of the Coimbra Laser Lab, and to thank the Coimbra Chemistry Center, the Portuguese Science Foundation, and the Remedial. So thank you. If you have some questions, please just ask me.